Chalo bachcho, let's see the question. So the question says in the PCR technology, the DNA segment is replicated over 1 billion times. This repeated replication is catalyzed by the enzyme. So इन चार options में से हमें वो enzyme identify करना है जो PCR technology में use किया जाता है. So the key concept in this question is to first understand the process of polymerase chain reaction, which is also commonly known as PCR. So PCR is a technique of DNA amplification. So with the help of PCR, we can make multiple copies of a single DNA template. Now the steps in polymerase chain reaction takes place at different temperature. Therefore, there is a thermal cycle which takes place in the polymerase chain reaction. So the temperature for each step varies in the polymerase chain reaction. Now, most of the steps in PCR take place at a much higher temperature. So most of the enzymes, so firstly we require a DNA polymerase for the PCR process to take place, because DNA polymerase is the enzyme which can synthesize a new DNA strand. So the amplification or the DNA replication can take place only by a DNA polymerase. Now most of the human DNA polymerases are sensitive for temperature. So they are mostly active and optimum at a temperature of 37 degrees centigrade. That is the body temperature. But since the temperature in the PCR reaction is much higher than that, therefore we need an enzyme which is more thermostable. So the thermostable enzyme which is used in PCR is known as the Taq polymerase. So Taq polymerase is a specific type of DNA polymerase, and this enzyme is responsible for the synthesis of new DNA strand. Also, the Taq polymerase is particularly used in the PCR reaction because of its thermostable nature. That is, Taq polymerase can withstand much higher temperature than the normal DNA polymerases, and therefore Taq polymerase is commonly used as a DNA polymerase for the synthesis of new DNA strands in the polymerase chain reaction. So now if we see the options, then the first option says DNA polymerase. So DNA polymerase is correct, but more specifically the enzyme is Taq polymerase and not the normal DNA polymerase. So therefore this is not the correct option. The second option is Taq polymerase. So Taq polymerase is the enzyme which is used in the PCR technology. And the third option is DNA-dependent RNA polymerase. So RNA polymerases are enzymes which are responsible for the synthesis of RNA strands and not DNA strands. So therefore, they are not utilized in PCR. So again, this is not the option. And primase is an enzyme which is responsible for the formation of primers. So again, this is not utilized for the synthesis of a new DNA strand, that is amplification of DNA during PCR. So the enzyme which is used in PCR is option number 2, that is Taq polymerase. So the thermostable enzyme Taq polymerase is the enzyme which is used in PCR technology. So this is our final answer. Hope you understood this well. Best of luck. Thank you.